This evening's discussion will be chaired by our General Manager, Sue Giddens, first one here on my right. Sue will interview the two dynamic brain researchers based here at the University of Auckland's Faculty of Medical and Health Sciences. Beside me, to my right, is Associate Professor Maurice Curtis, Director of Human Anatomy at the Centre for Brain Research and co-director of the Neurological Foundation Douglas Human Brain Bank. Joining Maurice is Dr. Malvindar Singh Bains, the Freemasons Foundation Research Fellow at the Centre for Brain Research. Our interview tonight will take about an hour and will be followed by a question and answer session. Please now join me in welcoming our speakers tonight.
Thank you, Nina, and thank you everyone for coming this evening. It's so thrilling to see a really full audience like this, and I know how easy it is to get to the end of a long day and think, oh, I might just have a glass of wine and stay home and watch television. So thanks for committing to coming out tonight. Got a really, really exciting interview happening tonight, and as Nina said, the first person that I'm going to ask a question of is Associate Professor Maurice Curtis, who has a long association with the Neurological Foundation Douglas Human Brain Bank. He's the co-director. He works very, very closely with the team at the Brain Bank, including distinguished Professor Sir Richard Fall, who was recently knighted in the New Year's Honours, clap, clap, and Marika Esej, who is the technical director and an amazing ambassador for the Neurological Foundation Douglas Human Brain Bank as well. So the first question that I'm going to ask tonight is a very obvious one, and it's one that we're asked often, and I know that probably Maurice and Malvindar are as well. What exactly is a human brain bank, Maurice?
Thanks very much, Sue. First of all, I'd just say hi everybody, and thanks for coming out as well. So I guess to kick things off, typically speaking, I would be teaching medical students in this classroom. That's the good news. The bad news is there's usually a test at the end, so I hope people are taking notes. A human brain bank really is a large collection of human brains that are donated by individuals either with or without brain diseases for the purpose of advancing medical science. And we're very lucky to have a large, well, we call it actually a boutique human brain bank, but compared to many human brain banks around the world, it is relatively large. And in the brain bank, we have brains from quite a wide range of neurological diseases. And so we have brains from Huntington's disease, Parkinson's disease, motor neuron disease, Alzheimer's disease, progressive supranuclear palsy, multiple systems atrophy, stroke, spinocerebellar ataxia, multiple sclerosis, brain tumours, and so on. And that's sort of the more common diseases, and then we have some rarer diseases as well that are represented in the brain bank. So really it's a facility that allows us to do a wide range of different types of techniques, different types of brain research. And in many respects, although much of the brain tissue gets used in this building, it is actually a resource that gets used in a number of different places around the world and across New Zealand. And I guess one of the things that is quite unique about our brain bank is that we do get a lot of clinical information. We get the background on individuals who donate their brains to science. And this means that the brain bank is not just the actual brain tissue, it's actually the history that comes with the brain. And that's really important when you want to study what types of symptoms people experience before their brain disease sort of kicked in or really before they understood that they had a neurological condition. So that's really what a brain bank is.
How does a brain make the journey?
Yeah, so the brain makes the journey. I guess it begins really with a discussion that happens over the phone or in person with an individual that wants to go ahead with brain donation. And that can often be years ahead of someone actually going through with the process of brain donation. And from there, we normally get some documents signed that indicate that it's someone's wish. That's sort of non-binding documents. Obviously, if someone doesn't phone, if the family don't phone us when the individual dies, then we won't know that the person died and that the brain may have been destined for us. But Then at the time that someone actually passes away, we get a phone call to say that mum or dad or the individual's passed away. And at that point, we kind of we get into a process that's reasonably streamlined, but it involves quite a lot of discussion with the family. Involves figuring out where the individual is, what the nearest mortuary would be, whether we can get the brain removed and then on its way to us at the brain bank. And this, I should say that we've had brains from as far north as Kaitaia and as far south as Invercargill, so we have been able to get brain tissue from all over the country. However, as the regional mortuaries have slowly closed down, there's less and less of them, it has become more difficult to actually get the brain tissue. That said, there's great cooperation from around the country to make sure this can actually happen. And so then a funeral director would bring the individual to the mortuary, the brain would be removed and then brought to us either on ice, or we would get the local mortuary to do the initial phase of fixation before the brain gets couriered to us here in Auckland. We had an interesting situation a few years ago where an individual passed away quite early in the morning in Invercargill. We phoned the mortuary and they said, yeah, no problem, we can do the brain removal for you. And then to our surprise, he said, and I can actually get the brain airside, and Air New Zealand can bring it to you on the 11 o'clock flight. And so I don't know whether if there's an Air New Zealand representative here, that might be a slightly different story, but nevertheless, the brain actually arrived in Auckland here shortly after lunchtime and became, a really valuable piece of tissue that we can use for research. So there's a huge commitment, really, from around the country to make sure that this can actually happen.
Excuse us, it seems in this day and age sharing a microphone's a little bit non-technological, really, isn't it? Malvindar, you study inflammation, or what you call neuroinflammation. I'm going to use some big words, but Malvindar's going to describe them, in neurodegenerative diseases like Alzheimer's and Huntington's disease. Tell me, what is neurodegeneration?
Well, can everyone hear me? First of all, we're testing the mic. Okay, fantastic. Neurodegeneration is actually a really simple word. You can break it up into two halves. So neuro equates to brain. And what we're thinking of are your main brain cells, or your neurons. So neuro, neurons, and degeneration kind of means progressive deterioration in this sense. So if you put it together, neurodegeneration is an umbrella term given to a set of diseases which leads to dysfunction and deterioration of your brain cells. Now, that means it's something that takes time. It's progressive. So conditions such as Alzheimer's, Parkinson's, Huntington's disease are kind of the main umbrella diseases that fit underneath this word neurodegeneration, but there are actually loads of them, and more are being discovered every day. So that's neurodegeneration.
Great explanation, thank you. So what does neurodegeneration look like, if you can see it?
Right, I'm hoping we have some illustrations here. But I guess there's three main things that a person, anybody, can visually see when they look at the brain of somebody who has neurodegeneration. So behind me we have an image comparing a cross-section, so a slice, of a normal human brain, one half of it, in comparison to severe Alzheimer's disease. So that's like quite end-stage Alzheimer's. And what you'll notice just with the naked eye is that there's a lot of shrinkage of the tissue, and we call this atrophy. So that's one hallmark of neurodegeneration. It's the shrinkage of the tissue. Another thing that can accompany it is we have a whole lot of spaces in our brain that actually delivers liquids to keep our cells nourished. And these caves, I'll call them, are known as ventricles. And in a lot of neurodegenerative diseases, these ventricles will appear a lot larger. So that's another hallmark of a neurodegenerative disease. And you'll notice in the image behind me, the dark black hole in the middle. Do you see that in the middle of the Alzheimer's brain? So that's an enlarged lateral ventricle. The other thing, I don't know if we have an image, we might do, is even though these brain cells deteriorate, sometimes they don't die off. And another hallmark of neurodegeneration, and typically in Alzheimer's, Parkinson's, and Alzheimer's is that the remaining brain cells might have a load of garbage within them, deposits and clumps of bad protein. And we don't know whether these protein deposits are harming the cells or are just being a byproduct of the disease process. But what we do know is that people who have diseases tend to have more of this. So it's almost like there's some sort of inability to dispose of the garbage in our brains in people who have neurodegenerative diseases. And what we're trying to do is get rid of it in easy terms. So those are the main things that we see as researchers.
Staying with Alzheimer's disease, why is the hippocampus area so important to researchers who are studying AD or Alzheimer's disease?
So I've been studying Alzheimer's disease now for 2 years, and I'm quite lucky as a research fellow to be funded by the Freemasons to do this kind of work. And a lot of us are focused on the hippocampus because, first of all, its location. It's in the temporal lobe in here, which is a part of the brain that's involved with our memory, our ability to learn tasks, and also remembering not just long-term memories, but also remembering people and places. And within the temporal lobe, this little seahorse-shaped structure called the hippocampus seems to be heavily degenerated in Alzheimer's disease. And that was one of the first things they discovered, is that this structure looks really, really small and tiny, and it's atrophic, so remember that tissue shrinkage I was talking about. So a lot of researchers have spent a lot of time focusing on this area because Alzheimer's is the most common form of dementia, and the hippocampus, being our hotspot for learning and memory, is an obvious place to look at to target therapeutic intervention strategies or, new drugs or something like that. And this is why the hippocampus is so important. But what I might add to that is you saw that image before. The whole brain is affected in Alzheimer's in various stages and in various ways. But the hippocampus, which I'm wondering if we can actually put an arrow on the hippocampus in the diagram, its location over there.
Can everyone see the hippocampus?
Can everybody see that?
Yeah.
And you'll notice in the diagram beforehand, it's basically a gaping hole. Do you see in the bottom I'd love to point it out actually. We don't have a pointer, do we? No. Oh, that's a bit of a shame. No, it doesn't. So what I want to hammer in is on the bottom, it would be my left, and everybody else's right. On the bottom of the Alzheimer's side, you'll notice a gaping hole. That's where the hippocampus was. And that's the reality of the condition.
Incredible, absolutely incredible. So obviously this is end stage.
Yes.
This is severe. So but this progresses.
Yes.
Yes, and that's neurodegeneration.
Yes.
Fantastic. Getting it. Maurice, let's go back to the brain bank now. How was it formed? What was the beginning, the creation, the formation of it?
So the brain bank actually really started in the 1980s, late 1980s, and Professor Richard Fall was working actually on the rat brain, and it so happened at the time that there was a group of researchers, actually genetic researchers, who were interested in Huntington's disease, and they had contacts with families that had Huntington's disease. But in those times, we didn't have a gene. We didn't know what gene Huntington's was affecting. We knew the chromosome. In the 1983, we found out what chromosome was affected, but didn't still know the gene. And so the only way to really be certain that someone had Huntington's disease in the end was to do a pathological examination. And Professor Richard Fall and others who were working with him got approached by families to say, can you confirm, in fact, that mum or dad or the relative had Huntington's disease? And so this required them to do dissections of the brain and to actually have a look at whether or not there was the characteristic degeneration happening there. And then the family said, well, look, actually, why don't you hang on to the brain and keep using it for the research that you're doing? And before long, it became apparent that actually there needed to be resources applied to this collection of tissue to ensure that it could be preserved long term and that we weren't sort of wasting, or that Richard wasn't wasting tissue. So in 1994, the Neurological Foundation gave the first grant to the Brain Bank, which was to buy a -80 freezer, which you can see there with Richard and Marika. And a -80 freezer, it's very, very cold. It stops a lot of the cellular processes that would normally degrade tissue from actually being able to degrade the tissue, and therefore it preserves the tissue long term. And that really was the beginning of the Brain Bank. So now we've got about 12 -80 freezers, and we've also got a lot of other storage space for brain tissue because the resource has just grown and grown. Yeah.
So you have tissue now from 600 brains?
Yeah, so we've got tissue from over 1,000 brains, but from whole brains we've got about 650. Yeah. So we do receive tissue from the neurosurgical theatres if people agree to give the tumour tissue or the epileptic tissue if they're going to have tissue removed to treat epilepsy.
Sorry about this. There's something very, very special about the Neurological Foundation Douglas Human Brain Bank that is very unique to this and very, very different to the rest of the world. Maurice, I'll get you to describe what that is, please.
So there's a couple of things actually that are quite unique really about our human brain bank, and one of them is really the connection that we have with families. So we actually do get to understand what the person lived with and then what the person died with. And that makes a big difference when you're looking at diseases that have a variable onset or way of onsetting. And so Huntington's disease, for instance, often people will have either a distinct mood problem or a distinct motor problem. Some people have a mixed scenario there. Other people in different diseases have different symptoms. And being able to trace those symptoms back to what's actually changing in the brain is actually really quite important because our brain controls our behaviour, and so being able to link those back is really important. The other thing that we have is relatively short post-mortem delays. So being a small country means that we're more likely to be able to get the tissue in quite a rapid manner. So I think our shortest post-mortem delay is about 2.5 hours. And the most recent paper I read that had looked at a large number of human brains had an average of 60 hours post-mortem delay. So that difference in terms of the quality of brain research is actually really important. So that link back to the families is actually pretty important. And if I could just mention one story, about 10 years ago we received the donation of a brain with frontotemporal dementia. It's the most severe and aggressive form of dementia that's out there. And we received the brain and did some work on the brain, and then 10 years later we actually got a call from a family member who said, look, we've had a discussion amongst our family. It was a large, cohesive family that really wanted to do something to help the work that we were doing on frontotemporal dementia. And so as a still relatively young scientist, I scratched my head and thought, gosh, what can we do with a whole family with frontotemporal dementia when I'm used to working with the brain? And actually it turns out that now we have quite a large research project on. We've involved psychologists, we've involved MRI specialists, we've involved molecular biologists and neuroanatomists to actually engage with the family and do a lot of different tests and studies that will help us understand the very earliest signs and symptoms of the disease coming on in these individuals. And to get a large number of people engaged in this kind of research is really one of the successes that I didn't think would have come out of my work and working with the Brain Bank. I thought it would be more with the brain. But actually that link back to the families has actually proven to be really critical. Now I do have I got a second to bring out something? I have a specimen that we prepared earlier.
Now this is real, so if anyone doesn't want to see it, maybe close your eyes.
So this is the human brain. And it's a little bit different to the brain that's sitting in your head because your brain is soft, whereas this brain has been plastinated or preserved in a particular way that it maintains its shape. But this brain that weighs about 1,400 grams, it wasn't actually one donated to the Brain Bank, it was donated for medical teaching, and so it's been preserved to allow us to teach medical students. But one of the things that's really unique about the brain is just how incredibly well organised it is. So there's a particular area that controls our movements, and there's a map of the body, almost exactly like if you were to take a photograph of yourself, turn it upside down and plaster it onto that part of the brain. That's the bit that would actually control your movement. And then all of your sensation, you think that you actually feel it out here in the periphery, but actually you're feeling it here. And if you took that same picture, turned it upside down and put it there, that would be where all of the sensing is happening from the periphery. And then thinking and intelligence, and then vision and control of your balance, and all of these things are controlled in very ordered, organised areas of the brain. One of the things that makes our brain bank really unique is that many of the brain banks around the world, and I don't want to say that we're the only one that does it, but many of them would take an approach that a pathologist would take, which would be to cut slices through the brain like this, take the specimens they wanted for the diagnosis, and then the researchers do the best they can with what's there. But you can imagine if you had 12 slices through the brain, there'd be a lot of wastage if you just wanted to look at a small area that's degenerating like the hippocampus. And so being able to actually block the brain, we actually block the brain down into very small slices, about 1 or 2 centimetres by a couple of centimetres. This means that when someone comes to do a research project, we actually take just that region from many, many cases and can use that. And so it's a very efficient, it's very efficient. It saves on a lot of chemicals and reagents to just deal with the bit of the brain that you're actually interested in. And this has meant that we've been able to do far more studies than we ever could have done if we'd just sliced going through the brain like that. We still do the pathology, so the pathologist still does an examination of the brain and tells us what the diagnosis is, which we can then feed back to the families. But throughout that process, by us giving the pathologist the blocks and we cut it so that it's prepared for pathology and for research, we kind of get the best of both worlds there. So it's about 1,400 grams, quite a large structure really for brains compared to a rodent brain, for instance, where most of the research is done.
Maurice, can you just tip it over like that so people can see the underside and then the 2 hemispheres? And can you just talk, which is the front and which is the back for the audience?
Okay, actually I probably should hold it like that. So this is the front, this is the bit that controls your emotions and intelligence and personality. And the area that Malvindar was just talking about is the temporal lobe there, which overlies the hippocampus. So underneath there and there, you have the hippocampus. And you can see the blood vessel, large blood vessels coming in to supply the brain there, and the spinal cord at the bottom there that's about the size of your little finger. So pretty important structures, those ones. Really, it's the frontal lobe we always talk about as humans that makes us uniquely human. But there's lots of other pretty important structures within there.
Just go like that and you can see the 2 hemispheres.
And then at the back you can see the cerebellum that actually contains almost 80% of the neurons in the brain, just right back in that area that controls our movement. Yep.
Remarkable. What does everyone think? Pretty remarkable, isn't it? So Malvindar, you use tissue for your research. Can you describe how you use it?
So we actually have a variety of techniques that we can use to work with human brain tissue. I'm a neuroanatomist, and what that means is that I spend a lot of time doing meticulous, labour-intensive work of studying individual brain cells in these brains. So there's a lot of microscope work. So I'm one of those nerdy scientists that sits behind a microscope and has a long look at the cells of people with disease brains compared to what we call neurologically normal control brains. Now, the way...
And you've got neurologically normal control brains in the brain bank?
Yes, we have. That's something actually I think we both didn't mention. So one thing about any study of a disease is that we're trying to find out what makes that disease unique. And how do you find out if something's unique? You compare it with the normal. Now, a lot of people think that, having a normal brain is not that useful for research purposes, but actually it's like gold to us. Because a lot of people only think about the idea of brain donation if there's a condition. And for us, we would like to know what makes that condition different from the norm. So I actually spend a lot of time looking at these brain cells in various parts of the brains. I guess you could say, let's take the hippocampus, for example. I might spend a lot of time looking at the hippocampus in Alzheimer's brains and compare it with control brains to find out what's different. So not just about what cells are dying, but also what interesting chemicals have gone up or gone down in these different types of brains. And from my perspective, I'm always interested in answering the following questions. What makes a brain sick? So the brain cells that are remaining, do they look sick? What are they expressing that's different compared to normal brain cells? What is spared? So what's still there? What about other disease features? What other disease indicators, or what we call biomarkers, are present in these brains that make them different from the control ones? So that's always what we're trying to do, because we're trying to find new drug targets here. So what's the best way to look for a drug target? Find out what's different in these brains. So one of the techniques we use exploits the properties of antibodies. Now, who knows what an antibody is? So when you have a condition, say you've got a cold, what happens in your blood? You get these antibodies that go up that are trying to fight off the foreign invaders. And what's really awesome about these antibodies is that they love sticking onto things. So we utilize the properties of antibodies to stick to proteins or cells of interest in disease brains to find out how they change compared to control brains. But there's something else I want to tell everyone. That's something we do in the tissue. But what's happening more and more, and what's quite exciting about our facility, is that not only do we have a brain bank, but we also have a biobank. And the biobank is dependent on the brain bank. So what's a biobank? What we do as researchers is we can take a small biopsy-sized piece of tissue. This can be from post-mortem brains, but it can also be from epilepsy cases in the hospital, for example, if they've had surgery, or tumor cases. And we can actually grow cells from the tissue. So essentially what that means is we can have the disease living in a dish that we can then test for new drugs and new therapeutic interventions on. So it's not just about getting the information from the tissue. We can actually marry that up and do functional studies from cells that have grown from these post-mortem brains. And that's kind of sci-fi, but it's actually incredibly exciting, because that means that any new drug target I find in the tissue, I can then talk to the people in neuropharmacology and say, hey, this protein changed. Do you have any drugs that could manipulate this protein? And if so, are there any cells growing from that brain that I was studying? And can we test that drug on it? So this is how fast research has gone. And that's just one of the other techniques that we can use. So as well as studying the tissue, complementing it with studying the cells from that tissue. Living cells, might I add.
Can you study then the mechanisms of the likes of proliferation and cell growth?
Yes, you can, and actually Associate Professor Curtis's lab does that, and it's quite exciting. They're trying to look at different ways to stimulate cell migration. And in addition to that, there's that aspect, but there's also studying other mechanisms. For example, how does that bad protein clumping stuff that I was talking about, how can we get rid of it? Or how can we make more of it? Or how can we exploit it? Is it a good thing? So there's so many questions that we can answer. For me, I'm interested in a phenomena called neuroinflammation, which is essentially the brain's immune response to a disease. And so, when you get a cold, a lot of things happen in your blood that tries to combat that cold. And likewise, when the brain gets sick, a lot of things go on in the immune cells of the brain and also in the blood circulation, which reaches the brain, that can be possibly exploited as a therapeutic target. So we've got so many avenues we can go on. For me, I'm looking at neuroinflammatory markers in the tissue, but I'm marrying that up with looking at neuroinflammatory markers in the cells as well. And we're doing a lot of work surrounding that at the moment because it's quite a hot topic.
It is sci-fi, isn't it?
It is. I never imagined, I don't know if everybody imagined that you could actually take post-mortem tissue and grow living cells from it. And this is the beauty of being in New Zealand with that short post-mortem delay and that high quality tissue, is we're working against the clock here. So the faster the tissue comes in, the faster we're mobilized to get to work to start growing something from them as well.
Outstanding. Maurice, you can look at different neurodegenerative diseases and stage them, I understand. And you have done this with a Parkinson's disease study. Can I get you to explain that?
Yeah, so there's a couple of things in this actually. So probably most people would think of Parkinson's disease, for instance, as being a disease that basically is a movement disorder, and that people have difficulties being able to initiate movement. They might have difficulty getting off the chair. They might have difficulty in starting a movement, but then once they're going, they're not too bad, although they might have a gait if they're walking, that sort of thing. But actually, one of the things that was discovered some years ago was that actually that is not the kind of end of the line. That's not the problem that actually begins the degenerative disease. And so we've worked on looking around in the brain to see where the disease actually shows up early on first. And one of the things that we've found is that actually in the olfactory bulb, you see the deposits of some of the key abnormal proteins in Alzheimer's disease and Parkinson's disease first. Now, this olfactory bulb is about 3 millimeters beyond your finger if you put your finger up your nose, which probably do it for homework rather than doing it just now. And the olfactory bulb, you can actually just, if you look carefully, in fact, I can hardly see it here, so you might not see it there, but there's a little bump there on the bottom of the brain. That's the olfactory bulb. And actually, it's the only, there it is sort of diagrammatically shown there with the little toothbrush-looking structure there with the little fibrils going down. That is actually where that is where Parkinson's disease begins. Those are actually the only nerves that are directly exposed to the outside environment. So in the case of your eye, you've got the lens in front of your retina. In the case of your ear, you've got the tympanic membrane in front of the vestibulocochlear nerve. Here you've got direct exposure of nerves to the outside environment. And what's interesting here is that this system is actually one of the few where you get regeneration in the brain. So when I started at university, it was believed that the brain had no regenerative potential. When I finished my undergraduate degree, I knew that there was. And the other thing that's interesting about this is that actually the disease starts and travels in the opposite direction from which the neurogenesis or cell proliferation actually takes place. So this paper here published in Science shows actually cells migrating down through that pathway there that we call the rostral migratory stream to replace neurons in the olfactory bulb. But actually the disease of Parkinson's and Alzheimer's starts in the olfactory bulb and tracks back in the brain, eventually affecting areas like the hippocampus and the substantia nigra. This work identifying, shortly after we published this work actually, the Fisher Trust funded some of the early work showing this retrograde travel of the disease back in the brain. But what's exciting about this is that if you have a heart attack or something like that, you've got minutes to hours to get to the, or a stroke, you've got minutes to hours to get to the doctor or hospital. Here you've actually got 6 to 10 years before the rest of the brain will actually be affected. And so it's quite a big intervention window. If we could find out how we could slow that progression down, then maybe that would be the way, rather than treating it when you've lost all of those cells further back in the brain that actually cause the movement disorder. And so that's very much what my lab is focused around. I don't know whether, I don't want to steal your question, Sue, but can I race on to the next one, which kind of leads on to that, on from that. And that is that we'd always been really interested in the idea of susceptible areas in the brain. At least this is what all of the literature said is how the disease spreads. So you have a particular area in the brain, the substantia nigra in Parkinson's disease or the hippocampus in Alzheimer's disease. And if you could fix that area of the brain, well, just the next most susceptible area of the brain would become problematic. And so you're not helping yourself much. You're solving the problem for a short while, but not actually solving the major problem. However, if in fact the disease spreads in the brain, then by stopping it early on, then you may actually be stopping it altogether. And this is very much the idea that we've been working on. And so some work that we published just a couple of weeks ago actually showed this very concept. And so this actually made it to the New Zealand Herald, which is very exciting. Yeah. And I think you've got a video there, Sue. So here you can see two cells that actually have something called alpha-synuclein in them. In the box, you can see that there's a red cell with a green spot in it. Now, this alpha-synuclein is a Lewy body. It's the problematic protein in Parkinson's disease. And what you can actually see here is that one cell that's containing green alpha-synuclein, we've made them do this, and a red cell that also contains alpha-synuclein but it's red, have actually exchanged that alpha-synuclein from one cell to the other. Now, we don't have all the images here, but this shows it most clearly. That cell will then go on to actually infect, if you like, another cell. And so this concept of a spreading disease in the brain actually has some plausibility, which means how do we actually go about studying this pathway to try and stop the spread of the disease? And we know that this alpha-synuclein or Lewy bodies seem to kind of seed one cell, and then a tree grows out of that, and then it seeds another cell, and so on. And so you get this progressive degeneration going back in the brain, and my lab is very much focused on trying to stop that. Now, using these cells that Malvindar mentioned from the biobank, we can actually see this in tissue that has been removed and cultured from individuals with Parkinson's disease. So we actually think that this is very much an active process in Parkinson's disease. Now, what we find actually is that many cells will actually chop up that abnormal protein and they'll get rid of it. Healthy cell will chop up the protein and get rid of it, and that's what's happening for most people most of the time, because there's a lot of these unusual proteins that the cell needs to get rid of. In Parkinson's disease, they are not so successful at chopping the protein up and getting rid of it, and so after a while, they actually just pass it on to the next cell, and that sets up the problem in the next cell. So this image, also from that same study, shows the tubes that the cell uses to actually pass these abnormal proteins from cell to cell. Now, they're called tunneling nanotubes. They're very small tubes, and they're like tunnels or straws that go from cell to cell. And this is a bit of a stepwise shift in how we think about the way cells communicate. Because usually when we think about neurons communicating, we think about them getting very close to one another and then sending a small neurotransmitter across the gap between them. The other cell picks up that neurotransmitter and sends an action potential, we call it, or a signal, down through the cell. But if cells are actually communicating with each other by having straws or tunneling nanotubes between them, it's a whole different way that cells are actually talking to one another. And one of the things that we've always had as quite curious in Parkinson's disease is why does the immune system allow all of these Lewy bodies and abnormal proteins to accumulate without scavenging them and getting rid of them? Well, of course, if the information is not passing outside of the cell into the next cell, the immune system never gets to see it. So it gets passed from cell to cell without the immune system being able to have a look at it and then scavenging it. And so we think this is a pretty important finding that we will certainly be following up on. It was about, I think, nearly 4 or 5 years worth of work. A very tenacious postdoc, spent about 4 or 5 years, Victor Derricks, working on this, but it was a great outcome, we think.
Thank you. Thank you, Maurice, and congratulations. When we look back 10 years ago, and this was Maurice 10 years ago, actually.
I don't have the elixir of youth.
Yeah, you were 17 at the time, weren't you? Yeah. So you had actually had travelled to Sweden, hadn't you?
Yeah, I was actually travelling. I was actually travelling I went overseas on a Neurological Foundation postdoctoral fellowship, the Wrightson Fellowship, which I was very grateful to receive. And on that particular day, I'm pretty sure it actually says it there, I'm guessing now though, March the 2nd, we actually found out that it was going to be published in the embargo came off just after I boarded a flight from New Zealand going back to Sweden, and it made the paper here, and then I got back to Sweden, it made the front page of the Gothenburg Post, and I landed just as the day had kind of ended, so I missed it, but we were quite excited by that finding. Yeah.
Yeah, amazing. It's every scientist's dream to be published in science, as you can imagine, and this young man to my right achieved it oh so young, yes, and was recently promoted to associate professor as well, which is another huge promotion, another huge achievement as well. Malvindar, we're going to talk about your research because you've recently been accepted for publication in a pretty big way. Tell us about that.
Yeah, so I think seeing someone like Associate Professor Curtis achieve his dream of getting, some sort of international recognition from, Kiwi-based research is something that all young scientists aspire to have. And the work I'm going to talk about took me 4 years, and it was my PhD work done under Sir Richard Paul and Associate Professor Henry Volvogel. And we've touched on Huntington's disease a little bit, but I just wanted to give you a little bit of background so that when we talk about the findings, it seems clear. And I'm hoping I don't see a single puzzled expression. And so it's not easy to summarize your PhD into 2 or 3 minutes, but I'll try my best. So Huntington's is a gene disease. So it means that if you have the condition, you were passed a gene from your mum or dad that gave you the disease, right? So you can pass it from generation to generation. So that's the genetic side. Despite the fact that it's a gene condition, the patients that have the symptoms which manifest in midlife often have variable symptoms. So, for example, some of them might develop an aggressive motor disorder. Others might develop cognitive and behavioral changes. So, for example, they might have mood disturbances, like they might be irritable, anxious, manic, depressed. All of these, it just sounds like me on a bad day, really. But, they have a chronic mood disorder. And so you've got a chronic mood disorder, you've got cognitive issues, behavioral issues, and a motor disorder, but they may not all happen at the same time, and they may not all happen to every single patient. And so once you start developing your first set of symptoms, and clinicians diagnose Huntington's by the presence of the motor aspect of the disease. So some of these patients might have chronic mood disturbances long before they get the motor symptoms. So one gene is causing all of these symptoms, right? The question is why? So researchers like me look to the human tissue to find out why on earth these patients, when they're alive, have such a dynamic disease, which makes treatment really, really difficult, and we don't really understand how one gene can do it. So in my research, I looked for 4 years. I'm not even kidding. This is not an over-dramatization. Associate Professor Curtis witnessed it. I looked at a region of the brain about yay big, okay? But I looked at it at so much detail. It's called the globus pallidus, and it's located, if I were to draw a line to the middle of my head from here and cut my brain open, it's a little structure virtually right in the middle of the brain. And it has subdivisions in it. And interestingly, some of these subdivisions are involved in our ability to create out a movement. So for example, me coming to pick this up, it has a role. But it also has a role in our mood, right? So what a great area to study. Why has no researcher done it? Because it's going to take a long time. And so my project was to study the globus pallidus in Huntington's disease by looking at brains bequeathed into the human brain bank. And so I studied 8 Huntington's brains, and I compared it with 8 control brains, and I looked at this area to see the amount of neurodegeneration in there. Now, we know what neurodegeneration is because I explained it's to do with the loss and decline of brain cells over time. So I was essentially looking at brain cells in the globus pallidus for 4 years in 3D under a microscope, painstakingly, for a very long time. But what was my motivation? Every single Huntington's family that came in and visited us, because they gave me a kick up the backside and said, get back to work, we need more answers. So long story cut short, the pattern of neurodegeneration in certain parts of the globus pallidus, right, mirrors the type of symptoms that those patients had when they were alive. So for example, a lot of brain cell death was found in the motor component of the globus pallidus. And these patients had chronic motor impairment. So the motor impairment scores were measured by a neurologist when they were alive. So essentially what I was finding is that the work in the post-mortem was sort of like a mirror image of what was happening to these patients in terms of their different types of symptoms. So the part of the globus pallidus that was involved in controlling your mood was chronically affected in patients that had massive mood disorders, but not necessarily in patients that had massive motor disorders. So I'm seeing nods, so I'm hoping that's sort of an example of what the research is that we can do. But what the hell does that mean? Who cares about the globus pallidus if it doesn't give us a miracle cure? But what it does tell us is that when we deliver that miracle cure, we can't deliver it to one part of the brain. We have to treat different parts of the brain differently. Better still, it means that this little tiny 2-centimeter area of the brain is really important for the disease. And that's actually a world first finding, believe it or not, nobody knew that. And that means that when people do MRI studies, they need to take what's going on in the structure quite seriously. And maybe we can detect earlier on, if the changes are going on here, what type of symptoms they're going to get as a consequence. So that means that we're one step closer to figuring out how this one gene can cause so many symptoms. And that came out from post-mortem examination. So that's the power of the study. And this was a science study accepted into a clinical journal, which means all the neurologists and neurosurgeons are taking this data very, very seriously when they're trialing deep brain stimulation in the brains of these patients with Huntington's disease. So I'm getting some nods, so it's never easy, but that was, 4 years of counting cells. Yep.
There's obviously something in the water at the Centre for Brain Research. What's the time? Look, we've got 10 minutes or so. I'm going to ask a really big question of each of you now. And I know that you could give me an hour-long answer, but we'll just keep it to a few minutes. For each of you, what is the most exciting aspect of research heading into the future for neurological disease? Maurice, I'll start with you.
Yeah, for me, there are a number of different aspects, but all the technology aside that will help us, get there. I still think that looking at the very early origins of brain disease is where the future will take us. So in 1958, dopamine was discovered. In 1962, there was a treatment that replaced dopamine for Parkinson's disease, and we've been using that treatment ever since. But that's still treating the substantia nigra, the area that's worst affected and that causes the movement disorders, once 80% of the cells have died. So you're still treating a disease that has had quite a bit of degeneration in it. And so for me, the big area in the future, and the area that we're really heavily focused on, is understanding the earliest origins of the disease and why it actually crops up to begin with. And then how do you actually stop it spreading from potentially the point of origin, not saying that it is, don't go away and think that something that you smelt 10 years ago is causing you problems now. But the olfactory bulb is where it crops up first, and so understanding what those initial changes are and then stopping it spread from there, that's very much an area of interest. The other area I think that is a hot area of interest but is looking a little bit aside from the neurodegeneration side, is looking at how cells actually connect with one another. So it sounds quite obvious the cells need to be connected, but actually one of the things that we don't look at all of the time and that I think is for the future is to understand how important those connections are, because quite simply a cell that isn't well connected just says, I have no reason for existence, and the cell actually gets chopped up and used for other things. And so keeping cells connected better, looking at connectivity in cells, I think, is the way of the future. It sounds probably quite trivial because things like exercise and diet and so on can help your cells connect, but obviously there's something in degenerative diseases that accelerates that deconnection, that the pulling away or the loss of connection between cells. So I don't think we're there yet with exploiting ways to get cells to connect better.
I don't even need the mic. So I guess, Associate Professor Curtis really hit the nail on the head there. We're no longer heavily focused on the cells that are dead anymore because we know that it's too late. So we're trying to find earlier and earlier and earlier interventions. And this diagram behind me, actually, the cell one, really summarizes how little we know, because all of these cell types behind me are in addition to our main brain cells, our neurons. And these cell types are the ones that we need to focus more on, mobilizing and getting them to do good things to help preserve our main brain cells or our neurons. And so I think a lot of the future work is focusing on non-neuronal cells, because the idea is to get these supporting cells to work better so that the main brain cells don't die. So that's sort of my area as well, and a lot of researchers are focusing more on the non-neuronal cells and also exploiting our natural immune system to try and do something to help our brains a bit more as well. Yeah.
Very good, thank you. I've got a photo here that I'd like you to explain, please.
Oh. I lost a bit of weight since then, actually. So that was taken 2 years ago at the Kiwi Bank New Zealander of the Year Awards, and I guess it was really exciting to be nominated and a finalist for Young New Zealander of the Year. I was actually a finalist 2 years in a row. Last year I lost out to Lydia Ko. So,
in good company.
I'm thinking I probably need to take up a sport. It could be what I'm lacking, but having said that, the New Zealander of the Year Awards was really a great opportunity to meet other people and also to really bring science into to the forefront of New Zealand interest. I think science and technology is the way to go to preserve all of our lives, and I think that's one thing I hammered at these awards. And you're probably wondering why I was nominated. It wasn't because of maybe the globus pallidus played a role, but really it's for a lot of the other things that scientists do to try and help support the communities impacted by neurological disease. And so Associate Professor Curtis and I are heavily involved in trying to support the families, and not just the families, but the associations of people, for example, the Alzheimer's, Parkinson's, and Huntington's associations, to try and use our skill set. So, for example, maybe we can give public talks on brain disease or how the brain works, just to give people knowledge and also prove that we're not hiding anything. And if you have a better idea than us, we want it. and we want to utilize it. And in the end, we're doing work for Kiwis. And as Kiwi researchers, we really want to give back in the best ways we can. So I think that's probably why I was nominated. But having said that, it was probably a good excuse to really push science into the forefront. And yeah, go girls as well. Yeah.
Before we get to questions, I just want to acknowledge that the two people sitting beside me here commit so very much time after hours to community service, like putting on public lectures and speaking in panel discussions and so on. They research, they teach, they have families. It's absolutely outstanding. And the undercurrent of what is behind all of this incredible stuff is jolly hard work and sheer determination and incredible passion. So I'd just like to say thank you to both of you. It's very, very inspirational. And we're very, very proud at the Neurological Foundation, that's for sure. So we're going to open the floor up to questions now. The confusing thing here is that we've only got one roving mic.
Oh, I've got one here.
Oh, you answer everything.
I can
Yeah.
answer.
Yeah.
No
Okay,
way,
so...
I'll take this off now.
Yes, Nina.
Very good. So when somebody is about to pass away, would you like the doctors or family to notify you in advance that their brains are being donated?
That's a great question, thank you. We haven't actually talked much about the process of donation. It's always helpful if we get a heads up that someone is deteriorating. It means that we can put, we can put plans in place to make sure that the mortuary staff, if it's over a weekend, I mean the weekends are the worst for us trying to deal with brain donation, particularly Friday afternoons as it happens. Everyone's trying to get home for the weekend and but if we can queue things up ahead of time, I have to say over the years, and Marika will, I think, attest to this, the staff around the mortuaries in New Zealand are incredible. They go well beyond what they have to. They don't have to do this kind of thing for us, but they always, if they can, willingly do it. We usually end up with a cell phone number and we just get told, oh, look, I'm not meant to, but just call me. So getting a heads up means that we can kind of prepare the way. We know we have that phone number that we can call if the person does deteriorate. If anyone's interested in signing up, then you're welcome, and Marika will just stand up for a second. Marika, actually. Marika is actually the star of the show, but she's our amazing brain bank manager, and often you might end up dealing with Marika during this process if you are interested, and she'll be able to give you one of these forms, which is kind of just an initial let us know that you're interested, and then we can be in touch. We actually can't accept all of the brains that we're donated, and this often is because logistically we can't. There might be the timing or something that means that we can't. The other thing is that because we are trying to understand specific diseases, we can't always accept brains from people who have got a mixed pathology, we call it. So if they've got Alzheimer's disease and a large stroke, that makes it very difficult we're sort of reductionists fundamentally, and so being able to tease apart what was stroke and what was Alzheimer's is actually a real challenge. And so we do have to be mindful of our resources and not accept all of the cases that we're offered, but we do try our very best. From here, we would actually talk to you or email you or whichever form of contact you'd best like, and then get a donor pack out to you, which you can fill in and send back to us. Then we've got the details on hand, and if you signal to us that your loved one is in decline, then we can sort of put the processes in place to see that it will most likely happen. We'll do our best to make sure that it can happen. Yep. Great question. Thank you.
Thank you all for coming. Thank you to our absolutely wonderful speakers. Thank you. And I know that there are a lot of fabulous donors here this evening. Thank you for all that you do. If you're not a member of the foundation, please see Nina and she'll give you a headlines issue and you can join up and get headlines and get first dibs on seats to wonderful events like this. Thank you. Thank you.